Hello viewers, I'm back once again, and you know my mission, and my mission is very clear: to create a symbiotic relationship between you the public, and the medical laboratory scientists for a very effective health. That is, we can always enjoy good health by listening and following the preventive aspect of laboratory medicine and general health. But the topic I have today is urinary tract infection. This has to do with the passage of water in the blood. The blood is filtered by the kidney. So it's the passage of that filtered water which comes out as urine, is that tract, that passage that we want to look. Infection usually happens along the line. It's either ascending from the lower down upward to the kidney, or is descending from the kidney down to the urethra. So from the kidney you have the passage called the ureter. From that place you go to the bladder, then to the urethra, and finally the waste is passed out in the penis or the vagina. But I am concentrating today on those infections very so popularly called by the charlatans staphylococcus, E. coli. These are names they have heard, but how do we come to ascribe that this is what is really causing your problem? It is not everywhere you find staphylococcus or E. coli that is the causative organism of any problem. So I shall expose it to you using the laboratory diagnosis that is commonly done with us as medical lab scientists, and I shall also expose you to the prevention, the reason why women are more prone to the disease. I have explained earlier, but I shall also explain further. So if you like what I'm presenting, if you like my presentation, if you like this video, my channel, just click the like button, click the subscription button, click the notification, and you shall always be alerted on when the next video comes out. Thank you for listening. Urinary tract infection, the laboratory diagnosis. From the picture we have here, these two are the kidneys, the ureter that comes down, it descends from the two kidneys to the bladder. You have only one bladder, and then the urethra. The outline for this discussion is to first look at the layman understanding, the federal government position, the worldwide view, the medical laboratory scientist perspective, and then the laboratory diagnosis, talking about culture, which is the gold standard, and the modern method of molecular diagnosis. The layman understanding can be understood from the perspective of what they pronounce the charlatans, calling staphylococcus, causing this everywhere. They cannot even distinguish whether it is STI infection, sexually transmitted, vaginitis, urethritis, or urinary tract infection. All they know is creating that confusion. They are confused, and you can understand why as we progress. Federal government really present it so well in a very organized way. We have the three health institution, the primary health institution, secondary and tertiary health institution. At the tertiary level, you find the nephrology department. You have standard laboratories that will perform all these various tests to treat urinary tract infection very effectively. At the primary level, it may not be that easy. It will just be urinalysis. But at the secondary and tertiary level, you have labs set up to really test all these problems. But I know these days I have seen some primary lab health institutions that have almost a standard laboratory. Research also departments accepts that the female are more affected, and that could easily be explained because of the openings, the orifices. The rectum, the vagina, and the urethra. These are all openings where bacteria settle. We know the rectum is where you find the feces, and the battalion of bacteria there, uncountable, can easily infect the vagina or the urethra. So that explains why women are very commonly infected. And understand too that women have short urethra, which predisposes them to urinary tract infection. The worldwide view accepts it's almost similar to the federal government position. It's the UTI increases in women, especially age 18 to 30. That's when they start having coitus and getting pregnant. You can understand. Especially in pregnancy, the positioning of the baby blocks the urine output, and once there is a kind of blockage, the infection is a very fertile ground for bacterial growth, the urine itself. So it's very easy to cause problems like the bladder problems, cause cystitis or other problems. We can also understand when women, postmenopausal women, have this kind of problem, because at that age the normal flora are no longer there. The normal flora in the vagina is usually the lactobacilli, but what you now find is the predominance of E. coli and other strange organisms that would now easily pass to the urethra and cause urinary tract infection. Diabetes too. At that age, many are diabetic. Diabetic helps the growth of many bacteria to grow. It allows bacteria to grow, to flourish very well, and so you can understand it will infect the urethra and cause urinary tract infection. The medical laboratory scientist's concern is very germane, because the urine is a very fertile ground for growth of all kinds of organisms, all kinds of bacteria. So how do they now define or really differentiate between the new turbidity, which is bacteria cause, that is the real bacteria that is in the system, and not the one that are contaminants? It's a very difficult decision. So the kind of urine collected is usually called midstream urine, and it must be freshly voided, so that the bacteria that you will find in the midstream, that is when you have urinated the first part out, which would discharge all the surrounding organism, then the middle urine, the middle portion of your urine, is what you now put in the bottle, the urine sample container. You put it there and immediately come to the lab. So the lab must act on it between 1 and 2 hours, so that the bacteria will not have germinated and grown to a very high level. Because urine is a very fertile ground, it will allow bacteria to continue to multiply. So how do you now know that it is not the contaminant that you are calling the organisms? That's why people shout staph. Staph is so rampant, it's so common. So when you see staphylococcus, you are already thinking it is the causative organism. Whereas it may not be, because the urine itself could allow this thing to multiply. So it is a serious situation where you bring your urine, your urine is freshly voided, and it's the midstream urine that the lab will quickly act upon to stop the continuous growth of the bacteria. The UTIs also could affect the urethra, the bladder, the ureter, and the kidney. So how do we differentiate the one that is affecting the urethra? The kind of urine you will collect for infection of the urethra, this area, is the first part of the urine, because the urethra can contain most times sexually transmitted disease. So it's that first part. The bladder is the terminal part of your urine.
where usually parasite like schistosoma hematobium cause damage there. So the last part of the urine is what is collected. The middle part is what you collect for the ureter and show anything that is affecting the urinary tract. So from that passage, what you want to collect is the organisms within that urethra to the bladder. So when the urine is passed out, the first portion contains problem with the urethra. That is, if you have STI, that can be used cultured to differentiate it from urinary tract infection. Then the portion of the bladder is the last part of the urine. So we must understand this very well. The specific isolate from the count, colony forming unit, and sensitivity testing of the isolate from the urine. These are germane questions you ask yourself, because when you have too many organisms growing, it now tells you that that must be the organism causing the problem. But when you have not done it very well, that is, you didn't collect the midstream urine, then there is a special count. We are looking for at least minimum of 10 to power 5 organisms before, when we do the culture, colonies, when we do the culture, to say yes, this colony forming unit is what is significant. We can say it is significant and it's causing the problem. So if it is staph that is significantly causing the problem, then we will say Staphylococcus aureus. If the species is aureus, then we say Staphylococcus aureus. But usually in women, it is E. coli, Escherichia coli, because E. coli is resident in the rectum, the anus. So you can always find it causing infection, because the urethra of women are short. So the urethra, the E. coli, is a very common significant organism in cultures that we do. We then do confirmatory laboratory test. These are the bottles. You look at the bottles, you have to label it. Your urine is designed just so beautifully well. The bottle, we call it universal containers, universal bottle. You can use it for both urinalysis tests or for culture as well. The microscopy, you examine the urine to know whether it's turbid or light, so that you can, from that alone, start knowing what to do. Because if it's highly turbid, like I told you, that shows there is infection. But when the urine is clear, then you are suspecting probably no infection. But there are situations where you have clear, sterile, something looking. To talk of content like the pH of urine is usually acidic. The specific gravity is just 1.01 and thereabout. The other things you will imagine to see are the protein. Protein is not supposed to be in one's urine. So the heuristics test for protein, blood, urobilinogen, bilirubin, and other things that will indicate the function of the kidney. It has very little to do with urinary tract infection, except when you start seeing nitrates and things that are connected with the pulse cells. Diagnostic continues with checking the microscope, under the microscope, wet preparations. That is, you take the urine, after centrifuging it, you allow the deposit to be placed on slide, just as done here. You can see the deposit is now placed on slide, and the cover slip is used to cover it. That will let you know. Then the stain slides are when you allow the deposit to be thick and you stain. This shows how it will look like after staining it. The microscope is being is being used to examine both the wet prep and the stain slide. The wet prep will show you things like this. You will see cells. When you see so many epithelial cells, it's usually in female because there is that area of vagina and urethra clashing. So you will see more of it. But in male urine, you hardly see epithelial cells, but very few. The pulse cells are indicative of infection. Once there are pulse cells is very indicative of infection. The blood casts is also a serious matter when you see casts that are showing red blood cells. The spermatozoa in the film usually can be seen in male or female early after coitus. The bacterial casts are also identified. It creates a kind of feeling that the walls of the kidneys are really being chipped off. But regardless, you report, but it's a sign that something is happening. The crystals are manifestation of probably a situation of dehydration, where you are having less water intake. The patient should be person should be told to consume more water, because this could form calculi and also block the urine flow, setting up more infection. Like I have said earlier, once the urine flow is blocked, the urine is a very veritable ground for growth of many organisms. The organisms multiply easily and find it. There are other things you will see which are done in this slide. HIV candidiasis is very common with HIV patients. You see candidiasis, and even with patients of diabetes patients. Trichomoniasis is another serious problem that, if seen, you want to do something about it. Then we go on to really culture the urine, culturing it on the Macroscopy agar or cleared, that is 16 lactose electrolyte deficient agar. That is more useful in a situation where you just want to use a plate. Cleared is very useful because it will allow the gram-negative and gram-positive organism to grow easily, unlike Macroscopy agar that may inhibit the growth of some of these gram-positive organisms. When you use Macroscopy agar, you combine it with probably blood agar or at times with chocolate agar. But with cleared, you may just use it alone. Macroscopy will take care of proteus. Cleared will take care of proteus spreading, instead of it allowing it to cover all the whole plate area. But cleared will allow other organisms to grow that would not grow on Macroscopy agar, which would necessitate using 2 agar. So for economic reason, it's better to use cleared and just inoculate using wire loop. There are calibrated wire loops which are very, very necessary. The DSTV and eating agar could also be good. They are very good for sensitivity testing. You can see the appearance of your clear zones. All the clear zones are showing sensitivity. The disc contains antimicrobial agent. So the ones here that are showing resistance because the antimicrobial agent cannot clear them. But with this one, the microbial agent clears it. That's why you are able to see the inhibition zone. Culture and sensitivity control method. When you want to ensure that what you are doing is perfectly okay, you may decide to use pulp tubes. These pulp tubes are more or less tubes that have 20 mils already prepared down. You just compare it to the kind of growth you see with the one you prepare on your own. It's also commercial. The cutting loops are calibrated loops that are already embedded with certain type of organism. If the organism can grow on your plate, it's part of what you use to check the performance, how luxuriantly it grows. So it tells that whatever you are growing is within all the loops here are very effective. The battery bulk is also to check if the plate you have prepared supports that growth. Biotyping is useful when you want to do the biochemical sugar test to know the biotype of the organism isolated. Eventually, you may get to the serotyping. There is also the battery drop, where you use indole, especially for enterobacteriaceae, to confirm E. coli, most especially oxidase, for pseudos and MRVP and all the other biochemical things. These are tests that are done. The sensitivity testing, you need to standardize it by using McFarland equivalent turbidity standard, so that whatever grows are standardized. There is no reason for somebody to say this thing is sensitive and in real situation in vivo it is not, though there are other things that could affect in vivo sensitivity. The reagent quality control kit is also highlighted here, and the slide, quality control slide, is also highlighted. Analytical, you find a situation, modern system where you use analytical profiling index for identification for microorganisms. It helps to quickly identify. The method, the standard method takes 3 days. The first day you inoculate your plate, the next day overnight the next day you check for the growth. Those colonies you examine. By the 3rd day, then you do the second day that you have done the sensitivity for the colony isolated. The 3rd day you now read your sensitivity. So you used 72 hours. But with these modern methods of API, identification system, rapid system 2, or molecular diagnostics, these are faster ways of identifying. The fastest remains the PCR method, where you multiply the nucleic acid amplification. You really want to, first of all, extract the DNA from the materials you are provided, then you amplify it, then you use your electrophoresis machine to know, this is the electrophoresis machine, to know whether it conforms with what you are expecting to see there. The line that you see, that line will let you know that is similar to the organism of interest. That is when you now go further to do other things to now know to claim that it is present there, because it's a faster method. Just within 24 hours, you have, the next day you have concluded the identification of the organisms and the sensitivity. That's also with the rapid method, the IDS method and the API. You quickly use the biotyping, the known biotypes anyway. These are organisms that you have already known, and it's so easy, unlike newer ones which you need to really do further studies on. So to speed up the time of getting results, that's why this method are being used. Faster method are used, but the standard still remain, the gold standard still remain culture and sensitivity. Nevertheless, these are helpful to quickly diagnose the urinary tract infection. Prevention remains what I've always said. Taking less of water, but in these days when E. coli that is highly resistant to fluoroquinolone then there is a serious problem. So one should more or less be careful, because E. coli is so common with women. From what we have said, the strain ST131 is becoming more frequently isolated from clinical cases, and it's highly resistant. So you can't imagine if one is not following the preventive method that has been itemized for all, which is so easy that you don't hold your urine for too long. You make sure you urinate whenever you feel the urination, so long as you can get a very near place to urinate. Then you drink enough water, so 2 to 3 liters of water a day, which will not allow your urine to concentrate, and you'll be able to wash away these style contaminants that, especially for women, it's around the vagina, because of the proximity of the anal region, the vagina, and the urethra. So the other aspect, since we have found out that between 13 years and earlier, as I exposed, the coitus problem that brings about UTI, if one urinates, it may help wash away this and reduce infection. So this is also recommended. Females should clean from front backward. The front is where you have the vagina, the back is where you have the anus. The anus contains so many organisms, uncountable. So you want to now advise that in cleaning after probably urinating, the right thing to do is to clean from front backward, and not back to the front, where you now chances are that you might transfer this resistant E. coli to your pets. If you find these things all I have shown very useful, just subscribe, and